that. So anyway, without further ado, please join me in welcoming Brent Mishler.
Can you hear me out of those speakers? Can you hear me out of that? No. Well, I got a different one. Oh, wait a minute. That sounds like it. It's channel 18 I think it's coming out of there. I can hear myself. Okay, good. Well, thank you so much, Paul. That's really kind. I don't know how important I am in the world of mosses, maybe. So it's great to be here. And I've always been interested in plants from several levels. I love them as outdoor things to look at. We've spent the last 2 days, Paul and I, looking around, looking at mosses. But I've always been interested, as Paul says, in how they relate to each other, how they relate to the environment, and how we can understand the evolution of plants. So I'm going to try to go through some more avant-garde ideas tonight, but I'm going to try to do it slowly and carefully, and we're going to pause halfway through to see how I'm doing. But the basic idea, I mean, the big intellectual question that's behind this that I've been working on for maybe 10 years is to try to understand why it is that the Mediterranean climate parts of the world, of which there's only 5, and we're one, only about 2% of the land surface of the world, is so rich in biodiversity. It rivals, in some ways, the rainforests of the Amazon, Southeast Asia. So I don't know that I have answers for that, but that's one of the driving questions. And I have at least studied all 5 of those areas. So we'll come back to that at the very end. But what so that's sort of the academic question driving it. But I'm interested in plant conservation and the urgency of plant conservation. And I'm going to talk to you after we have our short break about some new methods for better prioritizing areas that need conservation, sort of conservation prioritization. So it will go to more practical topics in a minute. The field that we're talking about here, it's called spatial phylogenetics. And if you're not familiar with what a phylogeny is, it's like a genealogy. If you take all the species that are out there in the world today, they all had some recency of common ancestry. And the phylogeny, what you're trying to do is put it together and figure out which ones are more closely related to which other ones, and hopefully how long ago. So for the general public, it's sort of like figuring out cousins, or even in this audience, if we went back a certain number of generations, we would find a common ancestor. So if we were going to work on that kind of question with us, we would trace back and say, you know, okay, it's 10 generations, I share one with him, and 12 with you. Okay, so like this degree of cousinness. So that's what a phylogeny is. And then spatial data, spatial data has been mobilized by getting all the herbaria digitized and a lot of citizen science. So we have tremendous amount of data about the distribution of organisms we didn't have 10 years ago. And due to advances in software and hardware, we can build phylogenies of huge chunks of life. So we can do a phylogeny for all of the plants in California or all the plants in North America, or we're trying even for the whole world. So the spatial phylogenetics, if you're familiar with GIS, which many of you are that are consultants, which is basically just putting information on a map, then what spatial phylogenetics does is boil the phylogeny down so that you can attribute it to each pixel on the map. And you can say what part is in each pixel. And then you can relate it to all the other tons of information, other layers of information that are available on the environment or soils or, human land use and that sort of thing. So that's what spatial phylogenetics is. It's a new approach enabled by the fact that we have great advances in data availability and great advances in software and hardware. So that's basically what it is. But let's see, the fundamental measure that I'm going to be talking about is called phylogenetic diversity. And phylogenetic diversity is let's say if we had a containing genealogy of all of humans, and we took the subset of humans that are in this room right now, and we looked at how closely we are related, counting back generations, as I'd mentioned, we could say, are we a random sample of human beings, maybe or maybe not. Certainly, if this was a wedding or something, you would have people really closely related who are in the room. But maybe if it's a big public event, you have sort of a random assemblage. But this is adding to the traditional way of measuring biodiversity, which was just counting how many species are present in a place. If you look online or you ask most people, what's biodiversity? They'll say something like how many species are in a place, which, that's a partial definition. But what this is adding to it is how connected are they? How far back do you go till you find a common ancestor? So in the phylogeny, which is one of these genealogical branching diagrams, the left one to be sort of like an example at a wedding where people are related, maybe in just two groups. If organisms are really closely related, versus on the right, the same number of species or whatever the terminals are, say 14 in both cases, so the number of species is the same, there's a lot more phylogenetic diversity on the right-hand one than the left-hand one. There's a lot more lineages, time, characteristics. And so we're going to keep coming back to that, and I'm going to explain really carefully how PD is calculated, and we'll have a chance for questions if it's not still clear. And I'm going to mention an additional metric, which you guys were talking about really small range taxa a minute ago. So there's a relative measure of endemism. Endemism has two meanings. Sometimes it means absolutely endemic to California or Humboldt County. But there's also a relative meaning, which is just the inverse of its range size. So something that's only in one place has high endemism, and some species that's everywhere has, basically zero. So endemism ranges from 1 to 0, where 1, you're only in one place, and 0, you're everywhere. And we're going to find, for the conservation purposes, we're going to want to focus on the ones that have smaller ranges. And I'll explain. Okay, so here's a cartoon diagram. This is way simpler than any phylogeny would be, but pretend we have 5 species in our global pool, but in 3 different communities. So each of these phylogenies, you see the trees are identical, but what you've done is had a different selection of 3 of the 5 terminals. So the one that's on the upper left, I contrived it so that I selected ones that have really long branches. The one on the right-hand side, I selected the ones with really short branches. And the one on the bottom is in the middle. So there's the species richness, species diversity is 3 for each of those communities, but the PD is quite different. It ranges from 90% of the tree to only about half the tree. So it's just adding up the branches. It's connecting the dots with common ancestors and adding up the branches. So that's PD. I hope that's clear. But then for PE, this is the exact same phylogeny. Oh, and by the way, the little numbers there is the percent length of the tree that branches. So the one on the far left is 25% of the tree is that one branch. If you take, that's called a branch length. So branch length in a phylogeny is how far you go back to the next common ancestor. So something like, let's say, a coastal redwood, you go back, I think it's something like 95 million years to the next connection to a common ancestor. That's a really long branch. Some of these other taxa, some of the manzanitas and ceanothus up here, it may only be 20,000 years till you get to the next. So that branch indicates sort of the time and also correlates with the amount of change that's present. So if we have PD, the way we get the PE, all I've done here is added numbers. The red numbers are how big the range is. So 1 is really little and 5 is big. If we take those original branches and divide them by the size of their range, you shrink down the widespread ones, you end up with something that to scale would look about like that. So we kept that really long, rare branch that's at the left-hand side. It's only in one place. It has high endemism, has high PE. The other branches shrunk down, but if we sort of blow up that area, you'll see interesting things happen, like in the middle of the tree there, the one with the 5 and the 2 in the upper left. The one with range 2 is a third of the length of its sister, whereas in the range-weighted version, since it has a smaller range, its range is almost the same as its sister. So the effect in PE is you're shrinking down the widespread branches, you're keeping the ones with small ranges, and then you're measuring PD on that tree. So PE is phylogenetic endemism, is just PD, but on a tree that you've adjusted the branch lengths to account for how common or rare they are. And that may not seem significant, but this is going to be super important when we get to conservation. There's a couple of derived metrics, which I have a slide to show you how those work, that are again measuring phylogenetic diversity, but they're comparing either the PD or the PE on the tree you start with a comparison tree. I call it the Lake Wobegon tree, where all the branches are equal. So where all the branches are equal, and then when you have RPD and RPE, when you compare the ratio of the PD on the original tree with that comparison tree, it gives you special information about when you have concentrations of long branches or short branches. And those of you who know these terms, paleoendemism and neoendemism, that's where that distinction is going to come in. Because when you have really long, rare branches, like coast redwood, that's a paleoendemism. So we'll come back to that. And then there's this method, which I don't want to go into too much detail, but it uses RPE. This is the one that identifies centers of neoendemism versus centers of paleoendemism. You're basically using RPE, and to digest it all down, you're finding places. There's a statistical test that goes with it, which involves randomizing all the occurrences on the map a bunch of times to see what your null would be. But you're finding a place like, and I'll show you in a minute, these forests here are centers of paleoendemism. They have a lot of rare, long branches. There are other places which tend to be more in the drier places, interestingly, in California, that have centers of neoendemism. And then many centers around here and also all the way down the coast have both neo and paleo. So I'll show you what that looks like in a minute. But right here, same tree, and I still am calculating PD in the same way. But you see that tree down in the lower right? That's the Lake Wobegon tree, that all the branches are equal. And so all I'm doing is I'm saying, what's the PD on my tree that I have in front of me that I analyzed versus what the PD would be if all the branches were equal. And that way, if the ratio is really high, what that means is there's a lot of long branches. And if it's low, that means there's a lot of short branches. So to make that real, we had an NSF grant that went for some years and produced an analysis of California vascular plants. This is not bryophytes yet. We have a brand new one on bryophytes. There's what the phylogeny is. I didn't put the names on the top. You can kind of see monocots, dicots there and things like that. And then these are the raw measures. So TR is taxon richness. That is the count of how many taxa there are in a grid cell. And PD is the phylogenetic measure. Weighted endemism is the endemism measure just using the species, not the branches, just how rare the species are. And then PE, I explained how that's calculated. And there's quite a bit of similarity between like TR and PD. And so what we have to use these spatial randomizations, the hypothesis test is taking all the occurrences off the map and put them back on randomly a bunch of times. And you get a distribution of PD, let's say, for a grid cell. And then you compare the real one with that distribution. And if it's way out as an outlier, then it's significant. So it's one approach to statistics that that is done in complicated cases where there's no known kind of parametric solution to it. So you don't get very much information from those maps right there, but check this out. So PD randomization is a test of whether the organisms that live together are more closely related than expected by chance or more distantly related. So to come back to our wedding example, the red places here are where the organisms that are in that place are closely related to each other as compared to a random sample. And blue places, which there aren't so many, but blue places are where the assemblages of taxa that are there are quite distantly related to each other. And you can address some ecological hypotheses with this. The case that's red there in all the drylands of California is explained by habitat filtering, where there are certain clades like cacti and legumes that are suitable to live in the desert. So there's an extra dose of them. Whereas when there is phylogenetic overdispersion, that's an indicator perhaps of a much older community where there's been time for them to sort out and exclude each other. If you remember Gauss's principle, from your general ecology, is that close relatives are not supposed to be able to coexist for a long time, that eventually one of them pushes the other out. So you tend to have communities that are structured by organisms that are spaced out a little bit in ecology. You can detect that with this if it's correct. And then RPD is an explanation that's about the branch length distribution. So you can see there's really long branches up on the north coast, really short branches in the desert and the south coast, and so forth. And those have various explanations, might be more biogeographic. Concentrations of long branches often indicate a refugium where there's relics of older floristic periods present. And certainly the areas that are in red are indicators that there's been a lot of diversification happening fairly recently. So you have a lot of short branches. So one way to visualize this in human terms is those centers of diversification are like nurseries where you would have a group of humans. If we were sampling humans all over the town, we'd probably find in the nursery at the hospital where the newborns are, we find a concentration of younger humans. And the blue ones are more like nursing homes, where if you happen to sample humans in some parts of town, you're going to find unusually older people living together. And then everything else is kind of a mixture in between. So that gives you some idea of what you're looking for, sort of cradles versus museums of biodiversity to some extent. And then I'm going to show you canopy results. This is not using PE yet, but the way canopy works, I've explained how RPE works, which is on the left-hand side. We have our original tree, and then we have that Lake Wobegon tree where all the branches are equal. So we're going to range weight both of those. We're going to range weight the original tree and range weight the comparison tree. And then in the original example, I did have a long branch in there and a really short branch in there. So we'll see what happens to those. And if you look closely, you'll see that the long branch is longer in the original tree than in the comparison tree, whereas the short branch is longer in the comparison tree than the original tree. So what that's doing is indicating those are the neo-endemic branches, and they're hard to detect unless you use RPE. This is technical, but if you just use PE, they kind of disappear because PE emphasizes things with long branches. It's literally their range. So these are all done. The analysis I'm showing you the whole state is gridded, and you decide what size grid cell by the data that you have. If you have sparse data, they have to be big grid cells. So these were 15-kilometer sided grid cells. And it's literally how many grid cells the taxon's in. Yep. And one other little concept I'll introduce with these cartoon trees that will come in later is everything we've been talking about so far is what's called alpha diversity. I'm sure you've heard that term before. Alpha diversity is the part of biodiversity that's just talking about how much is in each location. So you go to each of these locations, you say how much diversity is there, how much is there, how much is there. So that's one big component of biodiversity. But another big component of biodiversity is called beta diversity, which is how much difference is there between places. So California is famous for high beta diversity. We may not have as much alpha diversity as the Amazon basin has any one place, but if you go to different places, as you go up elevation or latitudinal gradient, we have really different communities in different places. So beta diversity can be measured phylogenetically in addition to counting species, and it's done by analogy in the same way as the PDPE. Instead of asking how many species are in common between two places, you say how much of the phylogenetic tree, counting the branch lengths, is shared or not shared between two places. You can do that pairwise, compare all grid cells to each other, and then do, if you know what a cluster analysis is, where you just sort of see which ones are similar to each other. That's turnover and that's beta diversity. And that's also important. I'm going to show you the canopy results. We're almost done with this part, and then I can take questions on these methods. The canopy results with the California vascular plant data are shown on the left. There is a very strong correlation with precipitation. Interestingly, in most cases, the places with the most narrow range plants are in dry places. You'd think maybe it would be wet places. The one exception is up here where we are, up in the Pacific Northwest. So you can see precipitation gradient. And then this right-hand diagram, what that is one of these turnover diagrams. So the branching tree-like thing that's over here is not the phylogeny, that's a cluster analysis of the turnover data. So two grid cells that are quite similar in their composition are going to be a similar color, and they're going to appear maybe close to some other things. So you see like the Channel Island taxa, pretty similar to each other. The taxa that are on the North Coast, pretty similar to each other. The next most similar set of grid cells is kind of the interior Pacific and so forth. So that's the turnover. That gives you an idea of how similar the communities are that are in these different centers of endemism. Everything that's colored, some color here, is a center of endemism, where statistically there's more of these rare narrow range taxas present than expected by chance. So I don't have time to go into all the details, but it's really cool. I mean, there's just all kinds of inferences you can make from this. And we're going to come back to the California results for the vascular plants and compare them with the bryophytes in a minute. In fact, right now, so it's not even published yet, but we have a new analysis that we did with several former students and collaborators, which is the same basic study as the vascular plant study California. The data is not as good, so we couldn't use the smaller grid cells because the bryophyte sampling is more poorly known than vascular plants. And we also did some tricks where we smooth the methods a little bit, which I probably don't want to go into, but for these statistical thresholds, you don't have to make it yes, no, significant or not. You can just actually use the probability in a smooth way. So these don't look quite exactly like the vascular plant ones, but the bottom row there, the phylogenetic diversity is on the left. The relative phylogenetic diversity is on the center, and then on the right is phylogenetic endemism. We never figured out what was a pointer hill here, Paul. Will it? No, not now. endemism on the south coast with the bryophytes and everything in the vascular plants. So the smooth, thanks. The smooth version of Canopy, we like acronyms, so we call that one SNAPE. So SNAPE is smooth neo and paleoendemism analysis. You can see that vascular plant results again in the lower left. And in the big diagrams here, there's mosses upper left, liverworts upper right, and then bryophytes together in lower left. And then an indication of where these grid cells fit in sort of the probability space. So I use my pointer here. So right in here, this is 0.95, which is usually the cutoff. So in an analysis like this with Canopy, you just said 0.95, that's it. Here, you're also looking at kind of areas that are close to 0.95. So 0.9 gets a little bit of color. So they're very similar, but what they do is they give you a little more nuance. The area where we are here is just super great, particularly with paleoendemism for mosses and in particular, and it shows up in this pattern. But the liverworts are the ones giving the signal on the south coast, not present in the mosses. That shows up when you put them together. And then over here on the east side of the Sierra, there's just like there is with vascular plants, there's a whole lot of diversity out there. Not so much diversity in the low desert. Bryophytes like the Mojave, but they don't like the Sonoran very much. We can go into why that might be. And then one last diagram, which I'll show you, and then we'll take a little chance for questions to be sure we're clear on all this. This is one of those ordinations. There's a cluster diagram there. The difference here with the smooth methods is we haven't made absolute cutoffs for what we're grouping together. This is like a continuous scale, but you can see some very strong patterns in the data. You see the California floristic province boundary here. And these patterns are actually kind of similar to the vascular plants in terms of which ones are similar to which. Once you have an ordination like this, you can do studies with the environment, looking at what are the environmental factors that are causing turnover and so forth. Okay, so I hope I haven't gone too fast. I haven't really got to the take home yet, but I want to be sure that it's clear. And go ahead and raise your hand if it's not. It's fine. These are difficult concepts. Go ahead and ask questions. Yeah, Gordon.
Yeah, you'll forgive my ignorance.
No, I'll totally forgive you.
I always do.
Slides where you had high diversity and low diversity, and you had all the branches, there's 14 major clades or something. I couldn't wrap my mind around which was high diversity, which was low. It's probably maybe the third slide you had.
Yeah, it's way back here, but we can go back. I didn't have that many slides.
There you go.
Remember the Zoomers can't hear or.
Yeah, Gordon was asking about this slide. Okay, so in the measure of species richness, so what we're talking about is some region we have 14 species, just some plot. We have 14 species. And what we're saying is, two plots, the left and the right, both have the same measure of richness. They both have 14.
Hello, Brent. This is Andrea on Zoom land. We cannot hear you anymore.
Now, yeah. Oh,
We can hear you again. Thank you.
okay. Okay.
I didn't touch it.
Oh, it's not your fault.
It turned itself off.
Yeah, the technology's, it's never human error.
So if you picture yourself, you could go to a plot in the Mojave where there would only be composites, legumes, and cactus. Whereas you go to a place here and every plant you look at is in a different family. So that would be, an example of the same number of items can have a greatly different value in phylogenetic diversity. Yes, ma'am.
How it's divided.
I'm-
You said it's the how long it takes to go from one to the other, but then it actually has all those values. Are they like years, or if you go on one of the others,
Yeah.
it's shrinking down.
Okay, so I'm going to partly beg off on this because my very first slide in the next section, I'm going to talk more about that. But the branch links in most phylogenies that you just see out in the literature, if you see a phylogeny of Cactaceae in the literature, the branch links are going to be in reconstructed amount of molecular change. And then there are ways to time calibrate that. So sometimes you see a phylogeny where the branch links are an estimate of number of years. So those are two different ways. And then that comparison diagram I was doing where all the branches are equal, that has its value too. That's called a cladogram, and that has value too. So I'm going to show there's a bunch of different ways. You've hit on a very interesting point, which there is a bunch of ways to measure branches. And it matters. It matters a lot. Yeah, I'll definitely go back into that. Thank you. Paul, you had your hand up?
Well, I think it would be helpful if you went to one of these maps that say has the canopy data for the vascular plants, and we were able to just compare some pixels that we know. Some places I don't know. This kind of thing. I don't really like the one on the right though, but so just look at the one on the left.
Oh, I'm sorry.
No, it's like try to look on the one on the left, right?
Okay.
So you can see where we are. So can you compare some close places where we are? Some that have paleo, some that have neo, and so on.
Well, the kind of dominant pattern up here and where we are is that mixed endemism where there is neo and paleo endemism. I don't know where you guys would know better than me exactly where those pixels are. This would be Klamath right here. Yeah. This would be like the Inyo White Mountains in Death Valley down here, if you've been there. And it's interesting for the vascular plants, there's a ton of stuff happening down in the Sonoran Desert. So that's a big difference with the bryophytes. There's paleo and neo also out on the Channel Islands. A very interesting area that does show up, and I'll show you again, are these northern Sierra foothills, like near Chico, all that private land that has the vernal pools and things. That's an extremely special area, and I'll be calling that out in the next part. But where we are right here is a very special area. You're living in a center of biodiversity. You probably know that already. Yes, sir. It's just matter degree. So super is like really significant. Yeah, it was just a cutoff, but statistics. So 0.95% is significant. And if you're 0.99, then it's super.
Okay, yeah, I know. So the red one, there's a red one, it's almost out in the ocean. And let me suggest that the red one is red because it's mainly right along the coast. And so I think maybe the red one is red is because you're like hitting the dunes or something, and there's loads and loads of endemics. And then if you just go, to a pixel that's adjacent to it, you get a blue one. And then that's maybe because it happens to get right in the middle of redwood forest. Would you
Probably.
think that would make sense?
Probably. But it's not the fact that it's on the coast. If there's less land area in one of these that's straddling the coast. But that would make it more likely to be significant. That'd make it less likely because it's very, its land area is small. I mean, this gets to this question about how big the grid cells should be. The answer is as small as possible. But so 15 kilometers is too big to answer your question. Think of 15 kilometers. A lot happens in 15 kilometers. So this is doing the best it can. You could do a much better study if you focused in it. And you guys know what ecological niche modeling is? That's another thing you can do to get finer scale resolution with some assumptions. What you do is you take your known occurrences and you compare it to these GIS layers that have, rainfall and day length and soil type and things. And then you fit a model to the points you have. And then you kind of project that and say, well, I don't know that this occurs there, but the environment's right for it. So you can have a range of attacks on that's embellished from the actual occurrences by, comparing the environments. So that's one thing that we do. But we need finer scale data to tell like the difference where you were out at the dunes to tell the difference between the open dunes and where the forest is. This data it's just too coarse.
I bet the red and the blue pencil are a field trip tomorrow.
They're right up there.
Big lagoon.
Big lagoon. Well, I'm going to show you your area here comes out really great in these next analyses. So let's postpone a little bit. And we did do a little bit of comparison with sort of a local map with areas here. Paul and I just did that while we were sitting around talking. Are there any questions about sort of methodology? These are really, really good. So I'll show you, I think the very first thing addresses your question. So what we call a facet, just like the edge of a diamond. So you can have one object that has many different faces to it. So what we're calling a facet is where it's the exact same phylogeny. The branches are connected in exactly the same way, but you've represented the branch lengths in a different way. These are the three main ones I mentioned to you a minute ago, where divergence would be an estimate of how much change occurred on a branch. If it's time calibrated, and if anybody here has worked with phylogenetics, it's difficult. It's quite controversial. But if you are able to scale it according to time, then the branches are representing years. And then the good old Lake Wobegon tree in the bottom, every branch is just equal. But the neat thing about that topology, if you have high PD in that, since the PD just goes up for every divergence that happens in the tree, having high PD on that is actually a measure of what evolutionary biologists call a center of divergence. So it's a place where there's been a lot of splitting apart of lineages, which is one of our conservation targets. So in this particular paper, which you can read all about it here, and there's a little app on the Jepson website, I think still somewhere that explores it a little bit. But what we are arguing is that we didn't want to choose among these for conservation purposes now. We're shifting out of pure academics to the kind of stuff CNPS is interested in, is how do we protect plants? That centers of important plants that are based on these three different measures have different value for conservation. They're not interchangeable. So often the goal in conservation, the stated goal is to preserve the maximum amount of genetic diversity in the world that we can. So if your branch lengths are based on the amount of change, high PD in that is literally the definition of high genetic diversity. That's going to be the place with the maximum amount of genetic change represented. If you're conserving based on a chronogram, then you're conserving kind of years of experience of the lineages have. That's kind of a weird goal. Maybe we don't have that too often, but that's just saying something like that redwood branch is 98 million years old. It's one of the oldest branches of any of our species in California. So that's something that's really. And then if you conserve places that are represented by pink here, those would be places where there's evidence that there's a lot of diversification represented. So these two are almost they're a little bit opposing to each other, because when we conserve biodiversity, if we only wanted to conserve genetic diversity, we would just take all the long branch taxa. But we also want to conserve the process of evolution. So if there's a place that's generating a lot of new diversity, we want to keep that too, even though it's one of these little nurseries where the branches are all really little. So I'm going to show you how this works. And just to show you how different those different facets are, for the California phylogeny, this is exactly the same tree, but these are the branches represented with those three different facets. So that's time. That's genetic divergence. And you see certain branches here are quite divergent from their relative. Often those are parasitic plants or something that have a very high evolutionary rate. And then here's the Lake Wobegon version of the tree, where everything's equal. So you can see how much difference that makes in this. This is what you were getting at. That makes a huge assessment of PD. But rather than deciding which one of these is the best, we realized after a while that we need to look at all three of them for assessing conservation. So now this algorithm that I've built up to that I want to explain to you, which may seem a little complicated, so I'll go through it slowly. In this case, the goal that we've set, probably you guys have heard of a gap analysis. So the old-fashioned idea of a gap analysis would be, they've been done for California a few times, is you look and assess species conservation across the state. This is a phylogenetic gap analysis. What we're doing is we're starting from the present day. We're using information from GIS layers about what is the protection status of land currently. We're looking at also at if there's intact natural lands present in a place or not. We have those distribution models that I mentioned. So here we're using finer scale data that we've done the niche models. And we're going to, there's our phylogeny, and we're going to try to prioritize all the pixels by which ones are the most important to conserve. So this is very practical, designed for agencies, CNPS, government agencies, Nature Conservancy, to help them in this 30 by 30 initiative and other sorts of initiatives. What we're basically doing, this is, if anybody here is into programming, this is a iterative algorithm that is starting with the map the way it is, and it's examining every single grid cell to see which one would increase the total value the most. Then it adds that into what's already protected, and then it iterates and does the analysis again for the next pixel. So you end up going through, there's 1,000 plus pixels here. You get a literal rank order of how important they are to conserve, like which is number one, which is number two. And what you're valuing would be a low current conservation status, a high intactness, which means it isn't under an agricultural field or a city, you know, there's at least natural vegetation there. And then high biodiversity value on all three of those facets, which means long branches in those facets, having small ranges. And then this criterion here, I'm sure people here have heard of the principle of complementarity in reserve design. When you're have money to have a new reserve, the way you decide what's the most rational place to have a new reserve is you look at all your existing ones and what species are present in the existing ones, and you pick your new potential one to be the most different as you can from those. So you have it's a triage decision. You just have X amount of money and you want to conserve the flora of this region. The next reserve should go in the place that's protecting the most stuff that isn't protected elsewhere. So this is using that same principle except phylogenetically. All this is being done with the branches. So you're trying to find the place that has the most biodiversity value according to those facets and is not very well protected, which tends to mean private land and things like land and things, but still intact. Okay. After we do that, I hope, does that algorithm make some sense? So it's a very objective, there's no human judgment in this. So I like the important plant areas program that CNPS has, and I wouldn't criticize it, but that has a lot of human judgment in it of people just, well, I really value that land. This is trying to get the human element out of it and just say, objectively, where are the places we need to conserve? And interestingly, check this out. So what this is, here's the same colors that we were looking at before. These are the 3 different facets. These are the top 50 pixels for each of the facets. Some of them hit in the top 50 in more than one facet, so they're colored with the overlap color. And the ones that are colored black, which look at where we are here, are the ones that score high in all of them. So they're centers of divergence, they have long evolutionary time branches, and a lot of genetic diversity as well. I'll zoom in on where we are right now, a slide that we just made last night for fun. But looking at this, you see, this is a very different picture than we saw with the unconstrained analysis of where's biodiversity in California. Like all the national parks aren't here because they're at a high conservation level. So they didn't make the cut. So there's the national parks here, there's Death Valley and Inyo Whites and so forth. There's Mojave National Preserve. So all the areas that are dark on this map, that would be places that are intact, but they have a fairly high protection level, or they're not intact. So you see the cities, and the Central Valley is also very dark. And these points are all coming out in the light areas. The light areas are the places that are intact, but poorly protected. These are almost all on private lands. So again, we see this wonderful area that's in the Sierra foothill here, I pointed out last time. And then also on the other side of the Sierra, a little bit up there by the, just north of the Central Valley. We have an area that my East Bay chapter has been fighting hard for. There's a proposed off-road vehicle area for right in that one. The Tesla, you probably heard of this result was introduced in that conflict as to why we need to save that area. We got centers of diversification on the peninsula, some down here in the south. So these are the places that would be not taking into account land value, because we could have factored that in, but we didn't. But these are the places that need conservation to conserve the flora. And I was, there's so much unprotected, wonderful area right around here. I was despondent about that, but Paul showed me last night what the North Coast Regional Land Trust has been doing. And all of these are easements or purchases in some extent. And I put this in here. This is the little region around where we are here that shows up in that other map. But it's so interesting that these are all filling in these places in here that, so very good. If anybody's here from North Coast Regional Land Trust, like, good job. That's really fantastic what's happening. So that gives you a little bit more chance to dig into these areas. This lets you more easily see what these represent. This isn't the canopy result, but still. And if you look at all the areas around the state that score very highly in this, all of them you'd say, yeah, that's obvious. That's super. The Mount Hamilton Range down there in the East Bay region, the Sierra Nevada foothills up here where we are. We did the analysis before Point Conception was saved, but we were super happy to see one of the highest priority places was right here at Point Conception. It's right where the Dangermonds gave the record amount of a single conservation donation to make the new Dangermond Reserve down there. You can't really go there yet, but you can drive through it. It's not open to the public. And I'm just about done. So when you do, here's the vascular plant results. And here's when we do the analysis for the bryophytes. The caveat here, this is only the chronogram facet. So it's only comparable to the kind of light blue ones here. But you see the patterns are very similar. in the North Coast for sure, the regions that would have the highest priority. This is using a cutoff, like 95% cutoff. This is using more the probability surface. So when you do this, you see it includes those areas, but it includes a few other shadow areas around it, and some based on those liverwort data, particularly down here in San Diego County and Riverside County, where there's some real global endemics down there. So the Brightfight results in general would support the vascular plant results, and we hope at some point soon. I haven't been able to get my zoological colleagues interested in doing this kind of thing. And I'm telling them if they don't do it, I'm going to take their data and do it. So one way or another, we're going to see what would happen with the vertebrates of California if we did this. Okay, so two slides with take-home messages. One, you can read there. It's counting species is certainly a very poor way. It's not nothing, but it loses a lot of the dimensionality you get when you add evolution. Interestingly, the limitation, and this gets to that grid cell size issue we were talking about, the limitation now is not actually building phylogenies. There's so much data getting into GenBank that we can build phylogenies almost anything. But now the limitation is having good distributional data of where the taxa occur. And I'm urging these to CNPS and I've given a talk like this to the Nature Conservancy and to try to get them to shift gears a bit. Okay, I want to return, being an academic, I wanted to return to my initial question, which is why the Mediterranean climate areas are so diverse. And I have to say, I still don't know. But I have a feeling, after having studied Australia and Chile and South Africa, and then the Mediterranean in Europe, that one thing that they seem to all have in common is that they have huge amounts of neo and paleoendemism, whereas other biodiversity hotspots in the world have lots of paleoendemism or lots of neo-endemism. But there's something about our environments here that stimulates generating new biodiversity, as well as sheltering pockets of older biodiversity that have gone extinct in lots of other places, like, say, coast redwood. And I'm guessing, this is all thoughts for discussion. I'm sure we have some good ideas here, but there's something about the disturbance regime, the patchiness of our landscape in space, edaphic differences and so forth, and then also in time with our incredibly unpredictable seasons that we have. There's something about that combination of environmental patchiness, which is just right. It's not like a desert. It's not like a tropical rainforest. It's somewhere in the middle. And so the factors have got to be in there somewhere. Fire too, fire and other unpredictable kind of disturbances. So I welcome any thoughts about these more biological topics or any questions about the conservation assessment methods.
In theory, is there generally like gradients of environment?
That's certainly environmental heterogeneity, like soil. It's also in places that have a lot of different kinds of soils, different elevations in a short distance. It's what you might expect, but what's kind of interesting, I'm not showing it here, but we've done some studies in California and Australia where if you're using those niche models as the way of making the ranges, then you can project them into the future using the climate change models. And you can say, well, what's going to happen to the centers of endemism that we have now in 50 years if climate trends continue? And what's interesting is that the organisms move, but like what you're getting at here is that these important areas still stay important, but for somewhat different plants. So, there's been this debate, what would happen if climate changed so much that Joshua Tree couldn't live in Joshua Tree National Park? Does that mean you get rid of the park because, Joshua Trees can't live there anymore? No, you'd want to keep it because even if Joshua Tree is moving north somewhere, then there's going to be things further south that need a place to go. And places like the Little San Bernardino Mountains have a lot of diversity. So yeah, so small scale environmental heterogeneity is really good for biodiversity.
Still trying to mentioning in the center of video and is enjoying here, and I'm wondering how much single species of redwood is driving the cell.
Could be.
Team,
Mhm.
you know, and it's why I'm wondering, is it just that they're so far both in time, but also in mutational difference from other species that single species is driving the cells up, or are they
It could be if they're really extreme, because the value you're looking at for the cell is sort of the ensemble property of all of them. So like maybe a gathering of human beings, if you have even 100-year-old person there, maybe it would seem kind of extreme to the random pool you have, but it probably would take more. So it all depends on what you're comparing with. So
if you have one person that's way more related to Neanderthals, and then the rest of them are aliens.
well, luckily we're all equally related to each other. None of us are more close related to Neanderthals.
That's for the second way you saying it, like we is driving the training that is running here, and it's also the diversity is showing more because we're actually at the right in between the very temperate climates, so we're both in the southern and south species range and the northern and of some other species. So maybe that's
Well, that's true. And but that's one of these turnover arguments you have to make. No matter where you are, you're going to be near the northern end of some species range and the southern end of a different one. So, you know, every place in California would have that attribute. But the turnover maps, if you're able to actually quantitatively look at this, then it's the places where a bunch of lineages are changing. So, you know, there, as you go south here and you hit a boundary where the color is changing a lot, that means the biota is changing a lot at that point. There's a lot of things that are dropping out, coming in. So these turnover analyses is what you would use to address that kind of issue of where are they're called biotic breaks. So where are the places where the flora changes like an unusually a lot in 15 kilometers? Yeah, go ahead.
I'm curious about stability in these ancient organisms, the orchid crabs, coastal redwood. Are you saying, I have two questions. The first is there a higher percentage or degree of these so-called paleoendemics in Mediterranean climates versus other areas of the globe? And the second question, totally unrelated, is are there any corollaries between and the marine environment and marine critters?
Yeah, needs to be done for marine organisms. I think that would be great to do. And then the question of whether there's particularly a lot of paleoendemics. The thing that I was mentioning is I think there's a unique mix of paleoendemics and neoendemics, but there are other habitats, let's say the Great Smoky Mountains, if you've ever been there, where there's a lot of paleoendemics, but not as many neoendemics. That's a very old flora. The Great Smoky Mountains is the only place in North America that's been available for plants throughout the history of plants. I've been under any kind of sea or any glaciers or anything, so there's a lot of holdover effect there. So there are habitat, and that's extremely diverse too, but it's just kind of diverse in a different way. And Amazon rainforest, parts of them are composed of older flora and parts are composed of newer flora. So by using the branch links, it lets you get at those questions, which you couldn't before. Yeah.
What data do you need in order to make these models?
Well, the basic thing is you need a phylogeny, and then you need a spreadsheet basically that has the organisms that are the tips of the phylogeny and where they occur. And so that could be a download of data from the herbarium database. You could also download iNaturalist data. There are data cleaning issues no matter what. For one of these analysis, you spend 95% of your time cleaning data and only 5% actually doing it, the fun part. And luckily, I don't have to do that part. That's like postdocs. So Andrew Thornhill, he spent 2 years of his life cleaning the data, and then we analyzed it in like a week. Because, you know, the herbarium data is messy. You got anybody who's messed with it knows there's older nomenclature, there's georeference errors, where if you look at the coordinates, it's got it out in the ocean and stuff. So you have to clean up localities and taxonomy, and the same is true with the molecular data that you're getting from GenBank. So there's a lot of data science to this, a lot of data cleaning. And yet once you have that, then it's pretty straightforward. There's some software, I didn't put in an advertising slide, but there's some software that's just called Biodiverse. It's Australian based, it's free software you can download, and it does all calculations you want to do with either species-based measures or phylogeny-based measures. Just Biodiverse. If you Google that, you'll hit it right off. Yes, sir.
Prioritizing this question is based on different methods of diversity. It sounds like it would create rather interesting museums rather than productive ecosystems.
Yeah.
Productivity needs to be factored in, otherwise you'll get something interesting information lists to look at, but maybe not a very productive system overall.
Well, for one of these facets, particularly the branch. And we could have a longer discussion about productivity, but productivity is I mean, if we just wanted the landscape of California to be productive, we just plant grasses everywhere, eucalyptus. So those of us who care about the native plants, who want to defend the native plants, productivity in an ecosystem, that's a very human centric kind of view. Certainly some of our opposition in Washington would love to do that. But no, what you're trying to do is you're trying to conserve the native flora, not necessarily the most productive plants that could live in the place. So the kind of production I think you're talking about is this evolutionary origination. We want to have, you know, because there is a lot of neo-endemism in our history, and we want to preserve that where, so I agree with you on that, is we don't only want to have museums of the older lineages. we want to preserve the cradles where new lineages are being generated. But I'm just saying productivity in terms of amount of biomass per day is not a great way to try to conserve native plants, because we're not as, grasses are actually more productive than chaparral. But I'm not saying let's replace chaparral.
But at the same time, for example, the Humboldt Bay is very good in terms of productivity as well.
Yeah, well, it's definitely showing up as an important place to conserve in everything. It also appears to be a region where there's also, you know, generation of new diversity and single old diversity. So yeah. Did you have?
Yeah, I'm just curious if endemism, like the extent of geographic range, is phylogenetically conserved.
Oh, that's a great question. So, you know, so the question is whether having small ranges is phylogenetically conserved, which would mean you would tend to have close relatives have similar values. And we've done a few tests for that. There's some standard phylogenetic tests. That's called phylogenetic conservatism that you can test. And most characteristics organisms have are quite phylogenetically conservative. So we were talking about cactus, you know, like you can tell cactuses, they tend to have the same kind of traits. And it does turn out that range size is one of these things which is largely correlated with phylogeny. Very interesting. But there are groups like, let's say, orchids tend to have small range taxa. And you could say, well, maybe that's due to pollination syndromes that they have or something. And there's other lineages, like those darn composites that tend to have really broad lineages, widespread ranges. So we're just working on that right now. I think that's a very important thing to add to this mixture. So far, we've been sort of agnostic about it.
So, Brent's gonna be around for a bit, so stay quiet and be married. And don't forget, there's all these other opportunities also, to spend time with him tomorrow. We'll talk about it. So let's give him a warm round.
Okay.
Yeah, I'll shut this down. You can sit there.
I'll hang around and be glad to chat more.
All right.
And or Zoom, long suffering Zoom people, you can probably let them go.
They'll take care of themselves.
Where's the